Here we are, last meeting before our first exam. And remember for the first exam, we're going to have assigned seating, so we can make sure people spread out and not sit right next to their best buddy. And so I'll have some name labels out and when you come in, and we'll get started right at 9:30, and then we have to finish up just a couple minutes after 10:45. So we'll use keep busy, which I have described before, and let's just start out by seeing if anybody has any questions they want to ask about anything at all. We can ask questions anytime, and just interrupt like that, okay? And whoops, I made a mistake. So the IP3 receptor and the ranitidine receptor are not exactly the same receptor. They have a really similar story. Last time I mentioned, oh, they're the same. They're very, very similar stories, but they are actually distinct receptors, just for your information. And both of them are channel receptors in the internal membranes that will release calcium, the second messenger calcium. And so that's that. Then what should you be doing this week? So we have our exam, and we meet on Tuesday. Then I realized I should just include the 2 pages of reading from the previous text edition that talks about the phospholipase C receptor pathway. So we've talked about it, we've said everything we want to say about it in class, but I'll just add that as a Brightspace announcement so you have direct access to it. And it's now part of our chapter 16 reading. It just isn't in our current edition. And then after exam 1 next week, we'll go to chapter 17, which is about the cell cycle. And I won't post it, and it won't be due next week, but just so you're aware that when we have our exam that day, I'll put a Brightspace announcement up and say, oh, we have some homework coming the following week. So a week from our exam, we'll have that drawing assignment due, and it's about the cell cycle. And so we'll be moving on after the first exam to our chapter about the cell cycle. We already included some of that, and so we're already leading into it. And then on to chapter 18, which is about programmed cell death, apoptosis, and some stem cell biology. Chapter 19, which is about cancer biology. Okay? And so I realized probably we should have another example of why there are signal transduction pathways. In our course, most of the time people just limit it to 2, but the third reason would be that there allows for big variety. So we talked about that last time and mentioned the idea before, that the same signaling molecule, for example, estrogen can of course cause different responses in different cells, like that. And that's really typical. And part of that is that the same receptor can activate different pathways in different cell types. So the target cell has already a pre-existing biochemistry. So when the signal arrives, that target cell is all ready to make a specific response, and it might be different than other cells, like that. Okay, so just that. So amplification, you bet. Integration, which is that crosstalk, that checks and balances, you bet. And then this variety, we can just get a whole bunch of different things going like that, okay? All right, so then here's something, and that is our course has one really big picture, and it's basically made up of 3 big pictures. And we've already been talking about the first big picture, which is about transcription regulation and about transcription factors. And how do you activate and inactivate transcription factors? How do you throw the switches to turn genes on and off? And there are more stories about regulation of gene expression than just the transcription step, definitely. But you know, this is a good one. It's the first one like that, and we've learned a lot about it. Then the second big picture is the one that we've been talking about, cell signaling, and all about receptors and signal transduction pathways like that. One of the reasons we're interested in cell signal is because if we activate a genomic response in the target cell, well, that's going to be probably involved in our first story. And so we just saw last time, oh, there are lots of receptors that activate different transcription factors. Some by phosphorylation, like STAT and SMAT. Some by stabilizing the transcription factor, like beta-catenin. Some by cutting the receptor, like Notch, and releasing part of Notch as a intracellular domain that becomes a transcription factor. So lots of different ways to turn on transcription factors, and that makes these connect. So some signaling molecules, like Wnt, extracellular, intercellular signaling factor, can activate gene expression in target cells through signal transduction and then regulation of a transcription factor, or some other story. And our third part, we've already started leading into, and that's the next chapter after the first exam, the cell cycle. And it all fits together because our favorite cellular response in our course will be this target cell decides, I'm going to grow and I'm going to go through the cell cycle. And we like that because that happens in our normal body. Whatever we learned last time, the bone marrow is producing 300 million new blood cells every minute, something like that, right? That's cell cycle, and it's just like that, you know? So we have a lot of cell cycle regulation that's really important, and cancer kind of always includes a story of, ooh, shoot, we got that cell cycle going, that was a mistake, like that. And so it's a good topic for our course to, you know, provide us a focus. You know, a focus example is, well, what about cancer and what goes wrong in these pathways? So they all three fit together. And in fact, that PDGF reassignment we just did, like that, the skin and the wound healing, and there was an example of a cancer patient in there who had a translocation rather, of the PDGF gene, like that. That's just our, you know, theme of our course, all put together like that. And, you know, we're kind of stretching because we're including some cell cycle stuff. But we can, you know, we already learned about that some in the first semester of intro bio, and you probably have talked about some other courses like that. So it's a good way to see, you know, that's what we're putting together. And we'll build up on that for our second exam, and then our third exam and our final exam will be other new material from other parts of our book. So that's our big flow, you know, kind of roadmap. And you could, you know, put it all out, you know, on the floor. All these different pieces fit together, okay? So this is just a quick screenshot of that PDGF receptor assignment, just to showcase my really horrible penmanship, you know, and I apologize, and I'm forever embarrassed about it. And I'm also bitter about it because in the summer of 5th grade, my mom made me stay inside while the other kids were playing and practice my penmanship. And so I feel bad every time I write. And, you know, my mom probably feels bad too. Right, you probably do too. But, you know, I'm talking about it, and, you know, so hopefully, you know, that it's helpful. But anyway, the point of this was to show it's a big picture. It all fits together. All the stuff we talk about fits together like that, you know? Okay? All right. So then we're in the middle of this where we're like, oh my gosh, that MIC, and we'll just jump ahead to this slide. That MIC is, that cells should not make MIC. You know, it's just like one of these Achilles heels. If you make MIC, you're, I don't know, you know, you're going to stimulate the cell cycle, and is that a good idea? So many examples of how the cell's naturally avoiding making MIC. And we can overcome all those just by increasing, just overwhelming it like that, you know, just making a lot of this MIC, by having a really strong cis-regulatory region. And we really typically don't get that by like a point mutation. You're gonna have a really big change in the cis-regulatory region. And so we saw that happen with the oncogenic retroviruses, and we're moving through that later, okay? And so we're really in good shape to understand these changes. That one, straightforward, very familiar to us. It's the normal MYC transcription unit that accidentally gets joined, gets picked up by the retrovirus, and when it infects a cell, the infected cell just makes way too much MYC, like that, okay? And so really similar, next story, but doesn't have anything to do with the virus. It's just what happens normally in our body, and that is that chromosomes can get damaged, they can get broken, they can get fixed, and sometimes they get glued together wrong, and that's an example. And there's a type of cancer called Burkitt lymphoma, or Burkitt's lymphoma, it goes by both names, that we're going to talk about, okay? And so MYC translocations are critical genetic rearrangements, often involving chromosomes 8. Chromosome 8 is the location of MYC. So the MYC gene, there's chromosome 8, named just on the based on the link, and we all have a MYC gene, and it's at chromosome 8. There's a long arm, it's a very specific location. And so scientists and doctors could see these translocations before that we knew what the MYC gene was and where it was located. And what they would know is there are some patients who have a lymphoma, and when we look at their cancer cells under a microscope, we see they've had this curious rearrangement, this chromosomal translocation, this mutation, where there's been some alteration in chromosome 8 and another chromosome. And their chromosome translocations, the breaks are just random, like that. Yep?
So it doesn't necessarily have to be between 8 and 14, just 8 and any other chromosome?
That's a good question. So we'll see that it's always going to be MYC, and we already kind of know what's going to happen. We're going to have that transcription unit, the MYC, and the chromosome is going to get torn between the transcription unit of MYC and its cis-regulatory region. They get torn apart and actually joined together with another chromosome that got torn between a gene on it. And we can already predict it's not just any chromosome, it's going to be one or a couple specific chromosomes. The woman who had that very unusual kind of skin cancer, the chromosome break was in the PDGF gene, and it was with another gene that was expressed at really high levels in the skin. So it was with the skin collagen cis-regulatory region, and she started making lots of PDGF in her skin cells. That's a big mistake. So here, it's not any chromosome. There are actually 3 chromosomes that are seen, and we'll see that they all have the same effect. They're all the same story. So it's a specific accident that happens, and all of them are going to cause the MYC to be expressed at really high levels, okay? And so it says down here, this drives rapid cell proliferation, acts as a primary oncogenic event, like that, So we probably have to have more mutations like that, because hopefully these cells would die by programmed cell death, and be taken care of by the immune system of the patient, that you'd be able to take care of it like that, okay? So there's just a zoom in. Again, that's whole chromosome, and you can see that it's specific, and people have seen that for a long time. That's how they categorize Burkitt lymphoma. The procedure that's done to see the chromosomes like that, that's been around for a really long time, is called karyotyping, and that's a karyotype, and it's where you take somebody's cells and you squash them so the chromosomes are spread out. You can see them like that, and there are some famous examples of health issues that involve chromosome translocation, like someone who is born with Down syndrome has a chromosomal translocation. Well, it's actually a chromosome duplication rather than a translocation, so not exactly a translocation, but certain things. Even before they could see the DNA in the genes, they could see these large-scale rearrangements of chromosomes. So when you do a karyotype, it looks like this. It's a squash, and the nucleus has just been squashed, and the chromosomes spread out. But then they have apps that just line them up and label them, right? So longest to shortest, and you can see these chromosomes have distinct banding pattern. So they name that banding pattern, and they talk about the long arm and the short arm. and then these banding patterns. And so we're looking at chromosome 8. And the only time you can see chromosomes is during mitosis. So when you're doing a karyotype, it has to be in a population of cells that are going through the cell cycle, and then just a few of those happen to be in mitosis, and you can see their chromosome. The chromosomes are condensed and visible, like that. So could you do a karyotype on differentiated muscle cells? No, because they're not going through the cell cycle. They're not, none of the cells are in mitosis, like that. Could you do a karyotype on your brain? No. Could you do a karyotype on a brain tumor? Yes, because the cancer is going through the cell cycle, like that. So there are plenty of cells that you can't do a karyotype on. It has to go through the cell cycle, and it would just be during that mitosis that you see it, okay, like that. And so just another image of that concept, like that, okay? And so this chromosome break, so then we could draw it, right? Here's a thing, right, where it could be, you know, draw out these chromosomes, label them. We're going to have to do that some on the exam. This is from the homework assignment we just did about PGF, where it's described there's a chromosomal translocation, and darn it, by luck, by chance, not luck, by bad luck, chance, the chromosomes got torn right there, tearing apart those genes, and then accidentally glued them together and glued them together wrong. Yep.
So would cancer only happen if the transcription unit of MYC and a cis-regulatory region from another chromosome like came together, but not if it was the cis-regulatory region from MYC and then the transcription unit of a different chromosome?
Yeah, right. So that's something that's just, you know, to kind of mention it one time. When there is a chromosomal translocation, we're drawing a picture of the gene that's going to become a problem. There would also be the opposite gene, right? We'd get the PDGF cis-regulatory region joined with the skin collagen transcription unit, and that's not going to be a problem because skin collagen doesn't cause trouble. It'd be expressed in the wrong place. Where does PDGF get expressed normally? It actually gets expressed in the bone marrow, in the cells that are making platelets, right? Because platelets have the PDGF protein, and in that assignment, the cell is called a megakaryocyte. Megakaryocytes are where PDGF gets expressed. All the cells have the PDGF gene. It's expressed right there. We know it must have transcription factors and stuff to get it going. And it would, you know, if the person had that translocation in every cell of their body, they would start making collagen in their megakaryocytes in the bone marrow. And we're like, I don't know, that might make them, it's cause them trouble, but it wouldn't be our focus. Our focus is, mix and match is the one that's going to cause trouble. Yep. Yep. So that's just, an example of a translocation in skin cancer that we can go, we can add that to our knowledge of the PDGF receptor, PDGF signaling pathway, and how it gets disrupted. And you'd be like, that translocation caused trouble in the skin because PDGF is an important growth regulator of the skin. And we talked last time about MYC is in the bone marrow right now, helping stimulate that tremendous amount of new blood cells every hour, every minute, like that. And this is, if you get the translocation there and you start making too much MYC there, those cells are already responding to MYC, and you're just saying, hey, here's some more MYC, right, like that. And so that's what's happening here. And so, again, we're mixing and matching two genes, but it's only a combination of one of those, like I just described, that's going to cause big trouble. And that's what happens in this Burkitt lymphoma. Again, people call it Burkitt or Burkitt's, and named after this guy who discovered it in the 1950s. And it's, are we talking about all lymphomas? No. Remember when we were talking about breast cancer? Are ER positive breast cancers, is that all of them? No. So this is a fraction of lymphomas, and it's a fraction where all of the patients have, it says I think 90% of them have that MYC translocation, like that. So almost all cases of Burkitt lymphoma are characterized by dysregulation of MYC. And I'll just mention here, we haven't mentioned it here in this course, just a thing that you can see sometimes. So MYC is an oncogene. And so wild type MYC is good, right? We're using that right now. But this mutant can cause cancer. Right? So it's normal, good, you know, it's an oncogene, and it just means it has the potential to cause trouble in a cancer situation. And so we have, you know, obviously multiple alleles of MYC, and we have some that are just doing fine, doing their job, and then we have some mutant ones. And so sometimes people want to refer to, you know, the normal version, and they use a term, a kind of a lettering system that goes back to those oncogenic retroviruses. Right? And so back in the 1970s, they said, we gotta have a name for these host genes that are in the retrovirus that are causing trouble like that. And so they called that VMIC or BRAS, like that, for viral MYC. They didn't know, you know, they're just like, that's not the one in the body, that's that viral MYC. And from that same setting, they also referred to CMYC for cellular MYC, viral MYC, cellular MYC. So it's just an old-time designation. Again, we've left the virus behind, you know, but people will still use that CMYC, and they're trying to refer to the wild-type wild-type version. And so they just happen to do it here, and I just, you know, might as well add that in. They say are characterized by dysregulation of the CMYC, and they're just trying to say wild-type MYC. Like that, it goes back, you know, it's kind of an odd way to say wild-type MYC. I'd just be like, oh, well, why don't you just call it wild-type MYC, right? By one of 3 chromosomal translocations, which place the MYC gene under the control of an immunoglobulin gene enhancer, okay? So what we want to say here is, again, you know, only a handful of students have taken immunology. But what we just need to know is in our blood, we have red blood cells and we have white blood cells. And one type of white blood cell, and white blood cells are involved in our immune system, and we have a population of white blood cells called B cells. And the function of B cells is to make antibodies, and we make antibodies to bind to infections and stuff like that, right? So antibodies are proteins, and we must have antibody genes like that, so that's an antibody gene. There is a synonymous word for antibody, it's immunoglobulin. Let's call them antibodies for our course. And these antibody proteins get made in these B cells, and they make a lot of them. They're specialized cells that are factories for antibodies. So what can we guess about these cis regulatory regions? And that is really, really strong. And so that's what we're doing, and you'd be like, dang it, that's such a bummer, because we've got a translocation in a cell that is normally being stimulated to grow by MYC. And the translocation brings a cis-regulatory region of another gene upstream of that MYC transcription unit, and it's a gene that those cells are also expressing at really high levels. So, is Burkitt lymphoma found? It's a lymphoma, it's a cancer of the blood, of the lymphocytes. And what kind of lymphocytes? B cells. There are other kind of lymphocytes called T cells. Is this a T cell cancer? No. Right? Because we have the antibody genes in every cell in our body, right? Due to genomic equivalence. Does that translocation cause trouble in any other cells in our body? No. They are not built to express the antibody genes. It's only in the B cells. If a person has Burkitt lymphoma, we don't know right away if they have it as like a new mutation. So it's like a new mutation maybe in those stem cells in the bone marrow. Or maybe they inherited the mutation from one of their parents, and every cell in their body has this translocation. We don't know that. This could be a family that has Burkitt lymphoma running in the family, and the baby doesn't get the cancer immediately, because usually you take multiple different genes to get mutated. This causes trouble, but there's checks and balances like that, you know? So even if a person had this chromosomal translocation in every cell, they would only get cancer in B cells, because that's where that cis regulatory region is active, and it's really active. So, and the last thing to mention is just, you know, again, a detail in immunology course, not ours, but it happens that an antibody protein is made up of subunits, and there are subunits from 3 different genes that work together to make the antibody protein. So that's why there could be 3 different translocations. Just by accident, it's connected MYC with one of those. They all 3 have really strong cis regulatory regions, each of these different antibody genes. And there's the heavy chain gene and the kappa light chain, the lambda light chain like that. So that's why there could be 3, right? Back to that skin cancer patient, there was just 1 translocation that brought PDGF transcription unit downstream of the skin collagen cis regulatory region, and that's going to cause trouble. Again, in that cell, that skin cell is got the PDGF receptor. It's ready to respond to PDGF. That cell, that skin cell, is making lots of skin collagen. So make that mistake and you start making lots of PDGF like that, you know? So same idea here. Frustrating situation, a cell-specific, B cell-specific cis regulatory region just cranks up the expression of the normal MYC, and it just overwhelms all the regulation of MYC. Try not to make it, you know, try not to get the RNA polymerase all the way down the gene, make the RNA unstable, try to inhibit RNA ribosome translation of it, try to make the protein unstable. You just make too much of it, you know, and it overwhelms those downstream regulatory steps. And so that's the whole story, Burkitt lymphoma. And so one of the above described translocations of MYC is seen in 90% of Burkitt lymphoma. But these oncogenic translocations are not usually sufficient to cause lymphoma. Other mutations must also be present. And I just read into this because it's going to foreshadow something ahead beyond our first exam. So don't worry about this. These additional mutations include mutations of the tumor suppressor, P53, which interact, the mutation, the tumor suppressor, I guess they're referring to the gene. Oh no, it must be another protein that interacts with the really famous tumor suppressor P53. And we'll see when we start looking ahead that in those, after the first exam, we'll find, there's some other genes that code for proteins that actually are inhibitors. And that's what P53 is, it's an inhibitor. And so those are called tumor suppressors. And cancer can be caused by either a stimulation of growth or a failure to inhibit steps in that growth pathway. And so that's what's happening here. They're just saying what we said. Well, you know, you could have a mutation in MYC and it doesn't mean you're already, you know, you just boom, you're gonna get cancer like that, you know? Like that, yep. And and it says, but with the TP53 and P53 mutated, apoptosis is blocked, you know, so they don't suppress that, and oncogenic B cells are allowed to proliferate unchecked. They mentioned other tumor suppressors like that too. So, you know, you probably have heard in, you know, your regular life, cancer takes multiple hits. You can start getting going, and then you'll get another mutation, another, and things just get worse, like a kind of internal natural selection of the cancer to grow faster and spread and stuff like that. Okay, so that's a really good example. And then, yeah, this definitely, you know, test questions, draw, label, and briefly describe the genes involved in chromosomal translocation found in Burkitt lymphoma. So you got to know the name of antibody gene, right? That gets made in B cells in our immune system, in our blood, and that's going to have the cis regulatory region of the antibody gene accidentally joined, repaired to bring it to the MYC transcription unit, like that. You could draw, you know, could be asked to draw that. You could be asked to draw the chromosomal translocation. We don't need to know the detail. We don't really need to know the number of the chromosomes. You would just label MYC gene, antibody gene, and draw a chromosomal translocation. You know, that zoomed out view that a person could see in a microscope, even back in the 1950s when Dr. Burkitt first described the disease. Like that. Yeah. Okay. And it's just, you know, and it's a very specific cancer, right? Trying to figure out what's going on in cancer. And it has really fascinating details like that. Beyond this, there's some additional things where you're like, oh my goodness, this is really crazy intense stuff, okay? So that's the second example, chromosomal translocation. And you see it has a lot of similarity with that first story, right? You know, it's just, you know, we gotta get this thing going with this changed cis regulatory region. The third one is something that happens a lot of times when a cancer is progressing. So it's going to get worse like that. This person has some cancer, maybe it's growing slowly, but it's going to get worse. And it's called gene amplification. And what happens in this case is just the entire MYC gene actually gets duplicated. So we learned about that idea, right? There could be a gene duplication and you could get a gene family. That happens where gene duplication occurs, and then over a long time in evolution, you get a gene family. You know, you get different, slightly different versions. So this is happening just in one person, one patient. So we wouldn't say we're creating a gene family, we're just duplicating the MYC gene. And in cancer patients that have amplification of the MYC gene, sometimes a patient will have 100 copies. That doesn't normally happen, but in cancer cells really are just getting more and more out of control, and they start to fail to do a good job replicating DNA. And they start losing chromosomes and stuff, and all kinds of stuff, you know? And it can happen that they copy this MYC and copy it again, and then, you know, you just have many MYC genes. It's not that it's acquired a new, strong cis regulatory region, but if you duplicate MYC, well, you're going to make twice as much MYC. 100 times as much MYC might overwhelm those downstream instability stories. And so this is just an example to kind of draw that out, just showing from a, you know, a review. You know, we've been talking about translocations, you know, getting a different cis regulatory region. They're just crank up the one MYC gene, and gene application is just, it's still the regular MYC gene, the intact MYC gene, just got many copies of it. And each time you increase the MYC that's produced, it's normal MYC, it's the normal cis regulatory region, but just have a bunch of the genes. And remember, whatever transcription factors are activating MYC, there's always lots of molecules of those. So if you have more MYC target genes, the transcription factors will just bind to those additional copies and just be like up, 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 right? And so this is just another very logical, kind of cause and effect, kind of common sense story like that. You know, where you're like, okay, you know, I can see that could happen. And so this is just an article, MYC gene amplification is often acquired in lethal distant breast cancer metastases of unamplified primary tumors. So they're saying you've heard this idea that cancer could spread. Oh, this person had breast cancer in their breast tissue, and it has spread, it has metastasized. And when cancers do that, a lot of times they're now in this stage where they've made changes, and they're just making more and more changes. And it's saying here that if a person has breast cancer and they don't see any MYC amplification in the primary breast cancer tumor, if it spreads, they start seeing, oh yeah, they're just having all kinds of problems. And one of them is they could have this MYC amplification, to give you kind of a practical example of how, right, that amplification is not going to be like the first step, it's like a later step, and you're like, phew, you know, this cancer is getting worse and worse for the, you know, patient, right? Okay, so 3 examples like that, okay? And up to here, that'll be covered on the first exam, like that. So it's a good spot to stop and say, you know, we'll start talking about new stuff that'll be on the second exam, but do people have questions about anything at all? remember I mentioned that sometimes I get an email over the weekend from a student and I'll be like, I better try to tell everybody about this, you know? Like either, you know, it's a mistake or, I've got to clarify that, they, you know, I have a really misunderstanding and probably I got to get that to everybody. So be aware that I can make a Brightspace announcement, you know, not the day of the exam, but maybe Monday morning. And, you know, you get notified of those, but, you know, sometimes there'll be something where I'll be like, that's that. And, you know, if that's that, you know, that's on the, you know, you've got to answer it that way on the exam. And sometimes it's a thing where I better make a little video. And I'm not gonna, you know, put a whole bunch of new material in, but, you know, there could be a little of that. So just, you know, all throughout our course, be aware of that, you know, that I gotta share something for the exam, and I'll do it, you know, we'll say at least 24 hours before the exam, where I'd say, we gotta make sure you know this, you know, piece, and you gotta watch for that, okay? But yeah, any questions about anything at all? And just always remember that, you know, the first time I taught this course, it was only questions like that. You know, like, take a look at this book. Yeah? Yep?
Could you quickly say one more time, the Burkitt lymphoma, the cyst regulatory and the transcription unit, can you describe that one more time? 'Cause
Yep.
I was getting confused with like the B cell, the antibody, like
Yep.
the way you.
Yep, so let's do that. Let's draw it out like we'd have to draw it on the exam. So it's gonna be a chromosomal translocation, and we just know, this is Burkitt lymphoma 1, and we mentioned the detail that there are actually 3 different translocations that MIC can do, because there are actually 3 different types of antibody genes. But let's just say an antibody gene, right? You know, like that. So we would want to draw, we'd say, let's draw the chromosomal translocation. And we want to, well, that's kind of a dopey drawing of a gene. Usually you'd make a line, right? And so here's the Burkitt lymphoma translocation diagram. Label it, you know, like that. And so we'd draw this first gene, and we'd say there's a transcription unit, and the promoter, and an enhancer. And this would be an antibody gene. So, you know, again, even though, you know, not everybody took immunology, you gotta know that name of that gene. And then remember that even though I don't label it, I really expect you to be able to write down your answers. So that's the transcription unit, that's the promoter and the enhancer, because that's, you know, we're just trying to demonstrate all the stuff we learned. So that's the first gene we'd want to draw, and then below it we draw MIC. So that's gonna be the MIC gene, and, you know, promoter and enhancer, and, you know, label it up so we know, you know, what you're drawing. And then you just go like this. You just be like, yeah, there's your chromosomal translocation right there. And it's true, it was asked, you know, you actually are producing 2. You're actually producing the other one, but the other one is not the issue. Yeah, this is the issue, where we take this incredibly strong cis-regulatory region that is cell type specific. It's in every cell, but it's active in B cells. They're specialized cells. Their job is to make antibodies. They make a lot. And we join it to MIC. And MIC is already normally expressed at very low levels to stimulate cell cycle progression in these kind of blood cells. So the cell is poised to respond to MIC like that, you know. And it does, right? We just overwhelm all the downstream stories. Yep?
So the translocation can occur with 3 different chromosomes because 3 different antibody genes create that antibody protein.
Yeah. And so we could write in, just to kind of help clarify that, you know, why, you know, this story. So this is chromosome 8. So we can say that's chromosome 8, and it happens that the 3 antibodies are on 3 different chromosomes, and I'm pretty sure one of the chromosomes is chromosome 11, and I don't know the numbers of the other. But there would be these 3 different observable translocations. They're the same story. You know, don't get distracted. Just make sure you understand, like this clarification. Yeah, it's an antibody gene. You know, that's the translocation. And again, we don't need to know the chromosome numbers, you know. If I ask, like, what chromosome number is MIC, you should just go slash the tires of my Toyota Corolla, which is red and typically parked by the gym. Yep. Yeah, Liana, it's like a 2018 Corolla. Yeah.
We need to know the.
I said that another time in a class, and a student said, I have a red corolla, and I was like, oh, whoops. Sorry, buddy. Yes?
Do we need to know the chromosome numbers for the P?
No.
G?
No, that, you know, that would be lame, right? I mean, don't you think? Be like, oh, well, you know, yeah. Yeah, and so other questions, anybody has anything else, yeah?
On the example exam you posted, it says, one of the questions is what part of the ERB gene is mutated in tumors?
Mm-hmm.
And what part of the genes are changed to create the CAR transgene used in CAR T-cell therapy?
Yeah.
Do we need to know that?
No,
Okay.
no, and we will on, for the second exam.
Okay.
Yep. Yeah, and so, you know, that first, that old exam I post that, I try to do that for each course I teach, just so a student can see the expectation. What's this gonna look like, you know? But the material changes, you know, each time, and so you're not expected to be able to answer all those. And I didn't send it out as like a study guide to be like, this is a great way to figure out, you know, the answers to these questions, right, like that. And I also don't send out an answer key, because when I did that, I just found students who just kind of glimpse at the answer key and be like, oh yeah, good. It's really to kind of struggle and be like, yeah, right? We haven't talked about ERB, you know, we haven't talked about T-cell therapy like that, you know? And so no. Mm-hmm. Yeah, and totally ask questions about those, because some of the questions you should be able to answer, right? And, you know, I mean, that's how instructors make exams. They go to their old exams and try, like, I gotta get some question for this exam, you know, and I try to make good questions then, you know, so, you know, I could change the wording and stuff like that, but it is a good resource to, you know, deal with in that way. And I just send one out for the very first exam, just because you never had this class before, you know? Yep. Yeah, but make sure you look through that, process that, see which ones you go, oh yeah, we talked about Burkitt lymphoma, I should be able to answer this question. The wording, remember I described that, right? So we want to be efficient, right? So I might say briefly describe, I might say draw and label, like that. I might say draw, label, and briefly describe. You pay attention to the key words in the question and follow those, and don't feel you need to do more. You can, but you know, the danger not getting through the exam if you're like, oh, Burkitt lymphoma, gonna write for 10 minutes about that. It's 2-point question, you know, like that. So just be mindful of that. Yep. Other questions people have about anything at all? Yep.
Can you go over like the PIP in the PI3 kinase again?
The PI3 kinase story? Yep. Yeah. And so the PI3 kinase. So what we should do is we should try to find FOXO pathway, but let's just go for it here and create what we kind of need to know here. But it's that FOXO figure, right? You're talking about PI3 kinase?
Mm-hmm.
Yep, okay.
2 to PIP3.
Yep, yeah. So, and we could, you know, have to make a simple drawing, so there's a cell membrane. And then we're going to have this receptor tyrosine kinase, which is a monomer, so a receptor tyrosine kinase. But it gets brought together as a dimer when it binds to the ligand, the signaling molecule that it binds to. And if we had to put a name on this, we'd say that's the insulin receptor, and that's insulin. It's a little bit more complicated than that. And when that happens, then we're going to get this autophosphorylation. Like that. And then we're going to have the PI3 kinase protein bind specifically to that phosphorylated form. And it's an enzyme. And its substrate is this phospholipid that has a long name, phosphatidylinositol, and we can just learn the name PIP. And it starts out where the substrate is got 2 phosphates on it already. So it's called PIP2. And the product of the PI3 kinase, the activated kinase, is another phosphate gets added. And so now we're going to have this as, I should draw kind of the same image, but it's going to be a phospholipid in the inner membrane of the cell, inner side of the cell membrane, and it's PIP3. And it's anchored in the cell membrane, so we talked about that. It's floating around, diffusing around, but just stuck in the membrane, so it's moving side to side, right? And a lot of stuff is anchored, RAS is anchored to focus that, activation there. So it's floating around, and it's going to bump other things, and so I'm just going to erase this and draw that PIP3. And I'll just draw this. We haven't talked about this, but it was mentioned in that, we haven't talked about this in class, but it was mentioned in that PDGF story. And I'll just show this, that anytime there is an enzyme, there is a kinase, it's going to take a substrate, and it's going to make a product that's phosphorylated. So it's going to add a covalent bond. And in this case, it's adding a covalent bond to that PIP2 to make a PIP3. So anytime there is a kinase, and I think there's 600 different kinases in humans because they have very specific substrates, they're an enzyme with specific substrates. Anytime that happens, there's actually also a reverse reaction to take the phosphate off. Almost any time that happens, there's a reverse reaction. And that's always done by a different enzyme, and it's a specific enzyme called a phosphatase. And the phosphatase would remove it and take it back to IP2, like that. And I'll mention that because sometimes this PI3 kinase is on a growth pathway. Sometimes it's going to stimulate the cell cycle. And so that pathway, when it's doing that, could get disturbed in cancer like that. So that comes up in later chapter. And we have uncontrolled growth. Well, the phosphatase that removes the third phosphate is called a tumor suppressor because it's doing the opposite, right? In some cases, this leads to a stimulation of the cell cycle, and as long as that PIP3 is around, we're going to be stimulating the cell cycle. And we could stop that by having this phosphatase remove the phosphate. And that's always the case, and so, from one example to another, we're usually focused on the kinase, and we don't bother with the phosphatase, but just because, it could come up with any of these examples. Oh, there's a phosphatase that removes it. And, and it stops it, right? It turns off this response, okay? So this is a response, and so what we've gotten PI3 kinase to phosphorylate the PIP, and we've got PIP3, and it's going to bind to proteins that are anchored in the membrane. And we said, we really don't need to know the names of those because there are a couple of different targets, and the targets have multiple subunits and kind of wild names. But once that target protein gets activated, it's going to activate a protein that we do want to know the name of, and that protein is called AKT. And AKT is a kinase, and when it gets activated, it's released from the cell membrane, and this phosphorylated form of AKT, so it's going to get a phosphate added to it, and that phosphorylated AKT is actually the enzyme that is going to phosphorylate FOXO. So that's a good enzyme for us to know that FOXO gets phosphorylated and inactivated by AKT. And so it gets a phosphate added like that. So that would be what we want to know from that signaling pathway. And again, we see that, we didn't need to learn the name of that protein. And you don't, again, if I put that on the test, or I'm saying like, why didn't you label that protein? Don't worry about that. But in other cases where we haven't talked about something, you know, you do have to learn stuff that's in the book, that's named in the book, if it's a prominent thing. Yep. Yep. Other questions people have about anything at all? Because we're not in a hurry. I mean, we are in a hurry to go on and learn more stuff, but we're also really focused on, if somebody wants to open a window, they can, because it's really overheated in here for people wearing coats and sweaters. And, yeah, all right, so we'll talk about some new stuff that's on the second exam. And you'll see, it's as we're going ahead, you're like, oh, this is stuff that we've learned some of this stuff's gonna be kind of familiar, right? And so here it is, ta-da, it's Herb B. So is Herb B gonna be our first exam like it was some other time? No. So Herb B is gonna be the last of these receptor tyrosine kinases we learn about. We're just gonna learn a little bit about Herb B. So again, we have 58 of these in the human body, and we learned about PGF receptor. We're gonna learn about one. We're gonna learn about one more also. So we're gonna learn about a total of 4 of our 58. PGF receptor, yeah, the insulin receptor, and then this Herb B is what we're gonna call it. Its normal name is the EGF receptor, and I just figure for us getting introduced to this, that sounds a lot like PGF receptor. So let's call it this Herb B. And Herb B was the name that those scientists gave the gene in the 1970s because they found it in one of these oncogenic retroviruses. And for the oncogenic retroviruses, they like to give 3-letter designations, and so that's what's going on here. Herb, and there's an A and a B story like that. So Herb B, and that's kind of I think it's kind of a nice one to remember. There's some gene, and when we say Herb B, we're talking about the mutated form. Either the one that was in that retrovirus or the one that's found in cancer today. Where there's no virus, it's just this Herb B, which is, like you'd say, the normal wild type version of it is called the EGF receptor, but we're just gonna focus on when it's mutated and causing trouble, it's still called Herb B. Okay, so that's our one, and here it is. So that's that RAS signaling pathway, that receptor tyrosine kinase signaling pathway. It could activate RAS, it could activate, what we just talked about, so forth, like that, right? But it's that familiar thing, but it's a different receptor, it's Herb B. And when it's mutated in cancer, it's a different kind of mutation. It makes the wrong product. So Herb B must have a mutation in the transcription unit. And the product it makes is a shorter version where it's missing some of the outside. And so remember how these receptor tyrosine kinases are monomers that come together when they bind the ligand. When Herb B is mutated, it's missing part of that, and it just makes the outside part sticky, so they just stick together. They don't need the signaling molecule, they just stick together. Now, remember we talked about how RAS could be mutated and stimulate the cell cycle. Well, this is upstream of that, right? It's earlier, and this receptor is just, this broken Herb B receptor is just always active. So it's going to be activating RAS or whatever signaling pathway and causing cell cycle control, right? And so Herb B fits into here. That's where we first got the name back in the 1970s. Scientists have been studying for a long time. And it's a mutation of the EGF receptor. And here's just an image of it. It's just showing, here's the normal wild type version of the receptor. It's got a nice ligand binding domain, and when it binds it, go, go, go. And the defective one is just got a weird, it's called truncated, shortened version on the outside. And it just happens that it's sticky. The amino acids are folded differently, and they just stick to each other, and it just turns itself on. Green light all the time, do, okay? So now we're going to talk, it's going to be a comparison of that Herb B situation, right? And just remember this idea, because you'll see we'll use this idea going forward in these next 3 chapters. that there's breast cancer, and some breast cancers, 70% of breast cancers are ER positive, and those are going to be really responsive to tamoxifen, because tamoxifen is an antagonist of the estrogen receptor. So ER positive means they're expressing the estrogen receptor, and if we could inhibit that receptor with the tamoxifen drug antagonist, we could decrease the stimulation of the cell cycle that's happening. And 30% of breast cancers are ER negative, and it would be a waste of time to give that very effective breast cancer drug to ER negative patients. That's a really hard time, a really hard thing, right? Where, a person gets breast cancer and they go to the doctor, and the doctor's like, oh, you have breast cancer. I'm like, well, my neighbor took tamoxifen, so I'm looking forward, to hope that will help me. And the doctor's like, we're not going to prescribe that to you. You have this ER negative breast cancer. And the patient's just like, it worked really well for my neighbor. Can't we just try it? And the doctor would be like, no, we're not going to try it. Like that, because it's a different deal. So there's the same story with HERB-B. There's a drug that can stop it, but it wouldn't stop other kinds of cancers because it's very specific. And the drug, I'll put the name up. The name we're going to give it is called the drug cetuximab. And again, it's not going to be a course in immunology, but it's a monoclonal antibody. So it's a drug that was made in animals that binds to the defective form. And I'll just draw a picture of what it's like when it binds. So here's that HERB-B, and normally its receptor looks like this, and it's only going to get brought together when the EGF arrives like that. So its monomers are going to get brought together. But then here's the truncated version, the HERB-B version, and those are just going to stick together. And you can guess what the antibody does. The antibody just binds right here and right here, and it just blocks them from being able to get together. And it's like, put your hand over it. You're like, good luck. Swear, swear, swear. So that's the drug. It's an antibody, and it's called cetuximab, and it just sticks and blocks that and inhibits it, okay? All right. And so, this is a kind of complicated drawing. Don't worry about it. It's talking about this thing, and this thing activates downstream pathways like that, right? And stuff. But here's that truncated form, and it just says these antibodies. And other companies have developed similar strategies because it's very effective to block that problem. So you find out, oh, the patient has cancer because they have that HERB-B mutation. Let's use one of these drugs like that. Okay. So that's a HERB-B, and we just want to talk about it a little bit. And you'll see that once we have that, okay, we understand an example. You can only use that cetuximab on a person who has a HERB-B mutation. It's not going to help a MYC or any other oncogene that's really focused on that. But you can see, oh, it's very effective because you just block it like that. Okay. Okay. So now. Just read down, this is just a Wikipedia page, ready? So it says, cetuximab is an epidermal growth factor receptor, that means an inhibitor of HERB-B, used for the treatment of metastatic colorectal cancer, metastatic non-small cell lung cancer, and head and neck cancer. Is it used on any of those? No, it'd be only used on ones where they detect that the patient has this HERB-B issue. So it'd be kind of like an HERB-B positive version of those cancers. There would be other cancers of the same type that just would not have that, and it would not be using cetuximab. So that's why I wanted to go back to the tamoxifen example. Same idea over and over again. This drug can be used on some metastatic non-small cell lung cancers, because some of them have the HERB-B issue like that, okay, right, like that. And, okay, so then let me read this paragraph because it's just crazy, right? It says in July 2009, so that's whatever, 17 years ago, the FDA approved cetuximab for treatment of colon cancer with wild-type KRAS. So we're just going to stop there and think about that. So the drug company creates this cetuximab and says, this is going to work great on ERB-B mutations. And we want the FDA to approve it. And the FDA approves it, but they say, you know what? If the patient also has a downstream mutation in RAS, this drug is not going to help that patient because the ERB-B problem is upstream of RAS. And so some patients that have the mutation in ERB-B would have a normal RAS, and it's not causing any trouble. And if you block the ERB-B, it will really help that patient. But some patients have a mutation that makes ERB-B, and they also have a mutation in RAS. If you use this drug on them, it's not going to help very much because you'll stop the ERB-B, but their RAS is rolling also. And so the FDA is like, wait a minute, you can sell that drug, but they're patients that you should not use it on, and you should not waste their money giving them cetuximab, okay? So see, in 2009, they approved it, but 17 years ago, whatever that is, they're like, already they're going like, we see, the molecular pathway, and we see that, it's okay to use this if RAS is wild type, but if RAS is causing trouble too, it's not going to be very helpful to use this drug like that. And see, that's one of these examples of like this cause and effect thing that makes sense like that. Every once in a while I say, like most of us don't know much about car mechanics, but all of us know that if our car doesn't start, you wouldn't check the tires, right? The tires are needed for the car to go, but not start. And that's this level of just simple logic like that. Like that, right? And so it's, fancy drug talk, complicated molecular mechanisms, but they're, the logic here is just that. So in July 2000, the FDA approved cetuximab for treatment of colon cancer with wild-type RAS. Okay? Since cetuximab had little or no effect on colorectal tumors harboring RAS mutations. So, you can draw that out, and it should be, it might take you a while to kind of sleep on it, but it should be like, okay, I got that. Because that's the next chapters are like that. They're going to be talking about cancer drugs, especially chapter 19, and they'll be just like, yup, okay. It's very much like the tamoxifen story. Only used on some breast cancers and not others. Don't even try it on others, right? FDA won't let you try it on others, right? Since it had little or no effect on colorectal tumors harboring KRAS. And remember, there are these 3 RAS genes, and that's that KRAS, so we don't need to worry about it. It's RAS, it's our RAS that we know about. Okay? Then this, 2009. This was the first genetic test to guide treatment of cancer. I can't believe that's true. It seems wild to say that. I think they had something going on, but the stuff we're talking about is pretty recent and more of it all the time. Then, check this out. So that was 2009. Then it says, in July 2012, the FDA approved a real-time PCR companion diagnostic test for KRAS. So, right, the drug company's like, oh my God, we want to sell cetuximab. We have to develop another test to test RAS to see if it's wild type or not, right? Because if we don't do that, they won't let us use the drug. So, they quickly developed that test. And that's this idea of being able to, assay what's the status? Do they have wild type RAS or do they have mutant RAS? And it has to be very specific. So, a lot of you students have done PCR. You're going to amplify the RAS coding region, and they're going to look for those mutations that would just change one amino acid. They're going to look for point mutations, right? It's a very specific assay they have to do on these patients before they are allowed to sell them setrusumab. So, you know, pretty recent and also pretty sensible stuff going on. And that's why we want to bring up this example. Just an isolated example, herb B, we're done talking about herb B, but it reminds us of a good example of what's going on, okay? Any questions about that? We won't mention it again unless you have questions, or maybe as an example of, you remember how they had to make this assay for RAS because they wanted to use their setrusumab drug on the herb B mutation, and they had to check RAS to do it. That makes sense based on the pathway, okay? All right, so that's that. And, you know, not on the first exam, right? It'll be on the second exam. So then we're gonna do that also with just one more receptor. Just one more receptor is gonna pop up, and it's called KIT. And we're not gonna look at it in hardly any detail, just a little snapshot example of KIT as another receptor, okay? And we're doing great. And so it goes to, here's a couple of slide figures from this important PGF receptor assignment. And the PGF receptor assignment, I described a way of assaying cells to determine how fast they're growing and dividing, and it's called the mitotic index. So that's an important number, and it goes to where, you know, you got a lump or a bump or a mole, and the doctor says, you know, we're gonna do a biopsy, take some of that, a little bit of that tissue and look at it under a microscope, do some assays. And an old time assay they still do all the time is to measure the mitotic index. When my mom passed away 7 years ago from old age, she was 87, and when she was like 85, she got some skin cancer on her scalp, and when she got that diagnosed, the doctor said, Lois, we're gonna give you some cream. It's not chemotherapy, it's just to keep your skin healthy up there, but we're not gonna treat that because this cancer is growing slowly. They're basing that on the mitotic index. This cancer is not gonna kill you. You know, you're already old, and you know, it's gonna take a long time, right, like that? So that's his mitotic index. And the doctor will come back and they'll say, you know, I'm not worried about that bump, I think it's benign. So one of the main things they're saying is, it's got a really low mitotic index. Or, I'm worried it might be malignant. And one of the numbers is, oh, it's got a high mitotic index. You know, and more there are additional tests. Is it ER positive, is it ERB, you know, like that. RAS, like that. They're more molecular tests, but this mitotic index is an old one. And so in that PGF receptor thing, I also talked about the mitotic index. And this is just a thing where they're using it to predict the survivability of dogs with a certain kind of cancer. And if it's they found, you know, if it's above this number, the dog has a, you know, a kind of life expectancy of several months. I'll try to just read that off. It says the median survival time, you know, sort of the average survival time with a mitotic index of less than 5 of these type of tumors was significantly longer, 70 months, than for those with a mitotic index higher than 5. Their median survival time was 2 months, right? 70 months versus 2 months, just based on the difference in the mitotic index, right? So it's a pretty important indicator. Here's something where these would be, yep, Chase?
For this exam or any exam, would we have to know like the exact mechanism for how the mitotic index test works, or no? Just know that.
Yeah, absolutely. And what you do is you look under a microscope at a bunch of cells, and you count them, and you count 100 cells, and you say of the 100 cells we just looked at, how many of those were in mitosis. And you can see that again because that's when the chromosomes are condensed, so it's visible. When you look at cells, normally you can't see the chromosomes. If you can see the chromosomes, they're in mitosis, so it's a very old measure. And I said 100 cells because it's a percentage. If you see 3 cells in mitosis out of 100 cells, it's 3%. Yeah, and that's mitotic index, yep. And it's an indicator of how fast the cells are going through the cell cycle. If they're going through the cell cycle faster, there'd be a higher percentage of cells that would be in M phase. What's the mitotic index in our brain? 0. They're not going through the cell cycle. What's the mitotic index in our skin? Well, it's slow, but they're going through the cell cycle because they're constantly sloughing off dead cells. What's the mitotic index of our skin under a scab? Higher, like that. What's the mitotic index of any cancerous cell? It's higher than normal, it's worrisome, because it's, you know, increased like that, right? So here they're just talking about that, and they're just using, and this would be a pathologist, so a pathologist is a doctor or, you know, a trained person who can make these measurements in the clinic lab and then talk to the doctor about what the prognosis is of this patient. And they're just making a table here, and they're just using two things. They say, well, how big was the polyp? Because they're looking at these gastrointestinal stromal tumors. So this would be an example where somebody does a colonoscopy and they're like, oh, we see some polyps and removed them. Sometimes they keep them and they do a biopsy. They look at them under a microscope and they measure the diameter of the polyp and the mitotic index. And they just made a table based on just those two things, the mitotic index in that polyp and the diameter of the polyp, and give the doctor the advice of how to do with this patient. So gastrointestinal stromal tumor with a polyp less than 2 centimeters in diameter and a mitotic index less than 5%, no need to follow up. That's like my mom, where they're just like, don't worry, Lois. It's, you know, it's not going to kill you. You're old already, you know, like that, right? And then, you know, very low risk, you know, the polyp is bigger, the mitotic is bigger, intermediate, high risk, bigger polyp, bigger mitotic index, like that, yeah? So gastrointestinal stromal tumor, we just want to mention that. It was talked about, described in that context, and we'll just finish up with this. I think it's the last slide about KIT. Well, we have one more slide showing KIT signaling pathway. Just this. So these gastrointestinal stromal tumors are the most common mesenchymal neoplasms of the gastrointestinal tract. GISTs remind some they are defined by tumors who, okay, and so this kind of tumor, it says these kind of tumor are defined as tumors whose behavior is driven by mutations in KIT. So that KIT receptor is mutated, and 85% of these have a KIT mutation, and then others have other kind of mutations. And look at this one, 10% have a mutation in PGF, like that, you know? So it's got something to do with growth response to a signal, like the ligand binds to KIT, or the ligand that binds to PGF receptor. And so I just read this one too. Down here it says, the identification of a molecular basis for GIST, that type of cancer, led to the exclusion of many tumors that had been considered as GIST previously, and also the incorporation of a much larger number of tumors that had been labeled as other types. So it's helping define what the cancer is. So we're using the assays not just to try to hope to find a treatment, but also to help us categorize these. And the last slide will be, you know, it's that pathway, it's KIT's that pathway. And so here's just a drawing of KIT, and KIT activating a couple of these signal transduction pathways. So really a familiar story for us, and it's just there's a mutation in the receptor that's causing trouble in this set of cancer patients. That's the type of cancer that those dogs have that could either be not worrisome because they have a small polyp and a small mitotic index. And so that's it with KIT. We can stop there. And I wanted to get that in if we could, just because that's beyond the first exam, but you can see how we're just rolling with our knowledge and building on it in this fourth section of the book, okay? So we'll see you on Tuesday, and we'll get started right at 9:30. And we'll have a nice spot for me to sit, you know, kind of spread out like that, okay?